So here I've got my whole poster. You may have noticed I went a little overboard with my markings connecting the processes, but it is what it is. So let's go ahead and start this. So we're going to start with our glucose here. And through the process of glycolysis, we're going to generate ATP along with some other products. So we all should be pretty familiar with this. We start with our glucose, and through a series of steps, we get our glucose 6-phosphate by losing an ATP. We lose an ATP, making this fructose 6-phosphate. And then we have a whole bunch of other steps here. What's important is right here at our phosphoglycerate kinase step, producing our 3-phosphoglycerate. We're losing an ATP and making an ADP, and also in the step before it, in our glyceraldehyde 3-phosphate dehydrogenase step, where we're producing our 1,3-biphosphoglycerate, we're also producing an NADH, which is used in reduction steps to help make our ATP. And then in this last step, in our production of pyruvate using pyruvate kinase, we are making an ATP. And I think I misspoke here. We're generating an ATP, not losing one. Now that we have our pyruvate, we can go to the pyruvate dehydrogenase complex, forming our NADHs here. And then those can be used to generate ATP itself, or further in this citric acid cycle, where at the end of this, we essentially generate a whole bunch of NADHs, which are used here in our electron transport chain, and eventually we will get to our ATP synthase and actually generate our ATP from ADP. So this is where we get our ATP, which is used as an energy source for the cell to build our building blocks. The prompt asks us, how do we make our nucleotides and riboses and how they come together to make ATP used for DNA replication. So if we go back to our glycolysis, in this first step, we generate our glucose 6-phosphate. And that glucose 6-phosphate is right here. And then that is used right up in here in our glucose 6-phosphate pathway. As you can see, it's right here. I have it linked to only my glyconeogenesis, but that's also another source from it after you've done the glycolysis. But anyway, your liver can make that. And then we go over here, and there's a whole bunch of steps, but what's important is that we generate this, our PRPP. That PRPP comes in over here when we're starting to make our nucleotides. Where are my edges on this video? Hopefully you can still see it. So anyway, this is where our CO2 comes in here, and it mixes with ammonia, and we go through this real long process until we even use that PRPP. And so we start with this CO2 that reacts with ammonia to make carbamic acid, and through a series of steps we're losing some of that ATP that we've generated, but as you can see here, we're also generating an NADH. And then we get down here where we're forming our formylglycinamide ribonucleotide from our formylglycinamide ribonucleotide right here. And, the reverse order there. That's also using an ATP, and also from our phosphoribosal amine to our glycinamide ribonucleotide, that is also using an ATP. So we get down here, and through another series we get of reactions, a lot of which use ATP, so we're using more ATP here. We end up with our inosinate. I forgot to mention that we also ended up with uridylate, and once we get over here to zoom out to our final step, you can see these are our precursors here to make our actual nucleotides. So up here we have another series of reactions, and our uridylate is going to be used to make our UDP, CDP, and TDP. You can see those steps here. I'm not going to go too much into it, but this step here is using an ATP, and to interconvert, those are also using ATPs, and that is how we get our UTP, CTP, and TTP, which will be used to make our uracil and our other ribonucleotides. And we also have our enosinate, which pretty much goes through the similar complexes, but there are two branches right here, starting with our enosinate. You use GTP to form this ATP pathway and vice versa, so that way you're not using GTP to make ATP and the exact same thing. And then that's how we get our ATP and our GTP, and those will be used to make the rest of our ribonucleotides.